Today I want to do a showcase of exploring the data, looking at gene expression and things like that. So far, what we are going to use today is the same data set we've been working on, but this is gonna be more hands-on rather than a click button tutorial. So the hands-on is gonna be like this. We're gonna do the scGEAToolbox and grab the non-QC files we have from the Becker data. So what we're gonna have is to open the SEEs, the raw data, and here I want to cover something I didn't do before. I'm gonna do union here. Might take some few seconds. All right, we get this. Now, instead of doing stringent QC, we are gonna do relaxed. And we are gonna see what's gonna come out now. It's keeping way more cells than before. And now, what I'm gonna do is the UMAP 2D. And yes, overwrite it, all genes. And I'm gonna come back in a few seconds. What we are going to do again is saving it in the workspace, and remember to close that. And we're gonna do the clustering now. Latent, we are gonna annotate that because it does not have anything on it. We are gonna have to put here 1.0 resolution, let it work, and gonna come back. Are going to open it again and get the object into that. And now it looks reasonable. This is the new clusters, and the UMAP looks better than before, has more cells than before. We're going to do the automatic human, yes, automatic annotation with Panglau. Okay, got it. And now it looks little bit more rich. For example, we have a lot of B cells, macrophages, endothelial, epithelial cells, podocytes. And what I'm gonna do here, because this is gonna be part of what we are going to do. Now we are gonna do hands-on. So what we're gonna do is save into the workspace again as SEE. Rename it instead of SEE1, SEE. Then save it, and here is where the hands-on is gonna begin. The first thing is that we're going to pass the cell type names with script. So what we're gonna type in here is SEE.C underscore cell type TX, and we are gonna get the unique of those, and this is gonna be cell types. And let's run that and see which cell types we have. My bad. What we have is B cells, endothelial, enterocytes, the very same. Now, what we are going to do is rename this one of the cell types. For doing that, we have to do IDX, find the cells that fit the condition we want. So in this case, it's gonna be from SE.C underscore cell type TX equal to, I want to rename epithelial cells. And let's see what's that. We run it, and oh, my bad. So we have to do semicolon in the end. So it doesn't do that because it takes time to load it. So IDX, and we are going to do sum. Okay, let me do it here. We're gonna run the sum of the IDX. Like that, we are gonna know how many epithelial cells were there, 900. And I'm gonna rename that as enterocytes. Then what we're gonna do here is SCE.C underscore cell type TX IDX, cell type Tx, IDX equals to enterocytes. Okay, this has to match. So what we're gonna do, it's you have to rename the IDX into getting the enterocytes, renaming the epithelial into enterocytes, and after we run that set of commands, we can verify that has taken effect. So we verify that here, and what we can see is enterocytes here and there, up there. And now we're gonna take out the all enterocytes. So we run again this code, and here what I'm gonna do is SCE2 equals to copy. That's required because MATLAB new style for objects requires copying like this. So we are gonna rename that SCE2 and verify that SCE2 has the things. Yeah, it has everything. And then what we are going to do is equals to select cells IDX, and then we run that section. Incorrect number of cell types input IDX sum. Okay, I have cells. Oh, my bad. Has to be SCE2 dot is calling object. SCE2 now has been subsampled. If we open this now, scGEAToolbox of the SCE, we can see that it's SCE2, sorry, not SCE, SCE2. We will see only the enterocytes, correct? And for the two batches, perfect. What we can do is differential expression here, but we are gonna do it by hand today, so we know and appreciate all this process. So the Wilcoxon, well, first of all, we need to take out the gene expression we want to look at. scGEAToolbox SCE2. I'm gonna try to, my bad. Let me copy that. Okay, first, let's look at the differential expression. This is kind of cheating, but shall be fine for the purposes. So what we're gonna do is differential expression, the new, use this one, yes, override, and it's gonna be by batches. Compare the two of them. It doesn't matter the order. We are gonna inspect ourselves. I'm gonna stop it and coming back. Yes, and whatever. Okay. And then it has to open the location. It's getting richer now. So no need for doing the enrichment analysis in the website. It's gonna be done here. Gonna stop it and coming back in a few seconds. Open the folder and we get the differential expression. Okay, looks good so far. I may just close this for now. And coming back to this, no, not that, the folder. The purpose of this is to find just one gene. We are going to use just one gene and explore that. I don't know, maybe, well, another thing we can do, we can just grab one gene and inspect the expression with Biolin plots. So plots, and we can do Biolin plot. Not here. Has to be SCE2. My bad. Okay, plot violin of batches. Sure. And it's gonna be gene expression. Paste. And I'm gonna paste. Well, let's copy a set of them. Let's copy all this stuff and let's select one. Let's go back. And okay. Yes. Here A and B. Gonna stop it and coming back. And I'll get it here. This is what we get. It doesn't look that differential as we might want. So let's look for, that looks fine. That looks fine. That's good, but I'm not gonna use that because I need something with some shape. AGRN looks like a good candidate. AGRN. See, and now we have different upregulation and downregulation in some of the genes. That's cool. That's cool. That's exactly the purpose. So we are gonna use AGRN. We are gonna grab this and closing it. And we are gonna extract that gene. Gene name, it's gonna be this. And that's exactly the one I'm gonna select. So I'm gonna look for SCE2.gene equal to gene name. This I'm gonna name it IDX gene. Then that's it. It's gonna be the index of that gene. Running this, and then that IDX_G. Oh my god. Find. In this case, I want to use find because I just want the index. Find, not the Boolean vector. IDX gene. My bad. Is the number 12. Okay, good. And we are going to export that expression. So exp gene equals to SCE2.x uppercase. Then I'm gonna put IDX_gene and all of them. That means all the cells. And we are gonna look if we're gonna show if it is or not. Exp gene, we're gonna do size. Come on. Size. Yep, is the number of cells. SCE2, and the number of cells is 2389 and 2389. We extracted properly the gene expression. Now, what we're gonna do is histogram. No, yeah, let's try histogram gram of exp g 20 beans, something like this, and plot that. Okay, we got it. And this is one of the problems of single cell, that it's very zero express most of the genes. And here's something interesting, that we have two batches here. So we can extract the batch such as this. We can do SCE2 dot C_batch ID. First, we can figure out what those are, unique, what those are, and we can call it batches. Then running it, and batches. Okay, we have those two. We can just copy and paste those, and that's what I'm gonna do for sure. Copying those and getting those here. And I'm gonna extract the the gene expression in the two batches. So it's gonna be exp gene A014 equals to exp _gene, but first I gotta get the IDX, right? So I gotta do it's gonna do se2.c underscore batch ID equals to that same tag name, right? And this IDX has to come in here. IDX. All right, let's do it. Should be around 900, something like that. Oh, if I run it, will work. Run and run. And let's look at that. That is size. Let's do size. Size. Okay, it's 1400. My bad. Now we do the other. Likewise, we can copy paste this, but replace a new tag for getting the other batch ID. And here we can call it CRC1. That's it. And let's do it. Now, size of exp underscore g underscore CRC1. 910. That's the one I was thinking. Now we can now plot those, and let me see if I can get the two of them together. I think it's gonna be like this figure, and then we are gonna hit hold on, and then print it again. Here we go. And instead of that, it's going to be the A0. Let's do it. Great. This is how it looks like. Let's increase the number of ins to something like 50 and 50 and see how that changes. Okay, here we go. Okay, didn't change much. That means I can reduce to something like 10 bins. I'm gonna use 10 and 10 and see if that helps me out or not. And that's it. We have that, the blue one. Which one is the blue? Okay, that's my bad. I need to identify which one is which first. But we can see clearly the difference, and that's why it's differentially regulated. Some of the population is within the 0.5, and that's basically driving the population difference. Now, if we want to do something cooler, we can do the kernel density estimation. After we extract the expression, my bad, let me close this. I want to point out from the website. Let me just close it. Website, kernel density estimation. We can estimate the density probability, and it's gonna do one kind of this density estimation, depending if you do normal density, KDE density, whatever. So you can just use it. So what I'm gonna do here, it's reuse my function, which is KDE. It's just as easy as this. But I'm gonna recycle this because it's gonna help us to have a nice plot. So we're coming back here, and I'm gonna walk through that piece of code. So this is one expression, and I'm gonna do that, and then the other. It's gonna do the kernel density estimation, and it's gonna get these two arrays. Oh my god, KDE. Position argument value not sparse. Must not be sparse. So here we have to do full because it says sparse. Full to convert it to full size, non-sparse matrix. Gonna do the same here. Full. That's it. Let's see if this works. Okay, it did it. And it did. It didn't. Oh, a parenthesis over here. Now it worked. Now let's look at what it produces. Two values. What those are, and returns f, the estimated PDF probability density function at the evaluated xf points. That means those gonna be used to plot. If we come back, we are gonna take now the mean values to see what's the difference of the density estimation, which is gonna be similar to the violin plots. We do this, and then we have mean 1 and mean 2. Let's look at those values. Mean 1, mean 2. We see clearly the difference, where CRC is upregulated. That's what we are finding. Now, we are gonna plot, hold on, and we are gonna do an area curve that has the density, the the probability density function and the points, and the given colors and shape and alpha and whatever. Okay, we got it. Looks cool. Now we are gonna plot the mean expression. What happened? Mean, is this working? Okay, that worked. Is this working? Okay, something happened, but it worked. Now we're gonna get this. This is gonna get the axis names labels gene 1. So here instead of gene 1, it's gonna be gene name. Gene name to get the name on the plot. And get the legends and hold off. Hold off means don't get anything else. Batch 1, it's gonna be, my bad. So we can code it here. Batch 1 equals to that. Oh my, oh my, oh my. Yeah. Batch 1. And here we can change it to batch 1. And here, batch 2. 2. Get this name. Here. Batch 2. Don't forget to run it. And run it. And then we can plot those. That's it. This is how it looks. We see a small difference here within these two numbers. And this is pretty much differential expression. We got differential expression, but in theory, this is what it happens. We test every mean expression and we can see how they differ in the shape in density. And they are clearly different. Where the cancer is widespread than the A014 sample. Cool. Now, question, how do I do the next? I know they are different, but for how much? Is this significant? Is it not? Okay, what we can do is rank sum. Wilcoxon ranked sum. Or the Mann-Whitney. And we have to do just as easy as this. P is gonna come from the rank sum of those. And we can just let in this. It doesn't matter which one is first. Well, it matters to tell upregulated, downregulated. But since we know what we have, we know which one has the direction. We run the Wilcoxon, and that's it. It says this much. If we look at the differential expression file, it should have a similar value. It's significantly different. That's it. What the ranked sum is, it sorts the values, right? Wilcoxon ranked sums. Let me see if I find something graphical. Wilcoxon ranked sum test. Let's see if we find image, because I think it's easy to visualize to some extent. Right, it's something like this. We sort it, and we test the median and mean, not just the mean, and tell, oh, it's by chance, not by chance that they are different. More or less, that's what we prove, that they are different by more than a chance. And that's it. I think I provided a hands-on work into how the differential expression works, how to visualize every single element, and do the Wilcoxon by hand. Now, all this is done in the code automatically for all the genes. We don't have to code it by hand, but this is just to know and be aware how's that happening in the back end. All right, nothing else to add. Thank you for watching.